Welcome to Synthetic Biology. I don't think our two speakers need an introduction. You've already had a good chance to hear some of their basic ideas. They were both on the project. Ken Oye and Michael Imperiale, speaking on synthetic biology. I understand that they have moved beyond a traditional presentation to be a dynamic demonstration of an interactive process, which can seriously kind of lead us to a new way of presenting this material that's going to be useful in the step beyond the report. How many people have this? If you need it, please raise your hand. It's about their presentation. Thank you.
Just hand it back and forth. Hand one back and forth.
Okay, well, thanks very much, and I really want to thank Notre. Whoops. That's okay.
Voice activated and floor activated.
And Don for inviting us out here. So when Ken and I were talking about how to set this up, and Don had originally envisioned that I would sort of present the topic, and then Ken would comment. We thought maybe it'd be better if we just sort of did a tag team, and we decided that we each know enough about each other's area of expertise to be dangerous. So I think that hopefully you'll enjoy this and that we can really get a discussion going here. So what we're going to do is really what's outlined on the bottom of this slide. We're going to talk about the state of the science and the technology, and then give an example of how dual-use technology has been dealt with in the past, and sort of do a little case study, and then use that as a segue into what sorts of things synthetic biology might be used to do in the future, and then take it back full circle into sort of the ethical and legal issues. And so wish us luck, because hopefully this is going to work out really well. So basically, synthetic biology starts with the ability to synthesize DNA. And over the years, there's been a need for doing this in terms of basic laboratory research. And the question is, can you get the gene you want, and then what can you do with that gene once you have it? And so Ken's going to talk a little bit about the technology, and then I'll come back to what's going on.
So actually, we are going to try to have this set up so that we'll mix it up. And Mike's going to offer some commentary on ethics towards the end, so we're going to try to confuse you as much as possible. So if we're talking about the technology, what we have up here is, I hope, Carlson's curves. And this is a set of curves. You're familiar with Moore's law. Well, this makes Moore's law look slow. Okay, you have exponentials on the vertical axis. And if you look at the different colored curves, one of them refers to improvements in sequencing DNA, another for improvements in synthesis. In other words, taking apart and analyzing DNA and figuring out what's in it, and recreating and synthesizing fragments of DNA. Now, again, those are pretty amazing curves. And I also want to tell you a little story. I was sitting around teaching at MIT, and big class, about 85 people. And so I was starting to go through this and talking about why those curves are moving so rapidly with reference to advances or changes in electronics and information technology. And scale, because you have larger and larger batches coming through and improvements associated with that. This hand went up in the back row. And the hand was attached to a guy who was saying there were other things going on. The improvements in sequencing DNA also resulted, and he was talking in particular about that kink in that blue curve where it starts going down even faster, to the application of modern industrial process methods, control methods. And he went on at great length. And after the class, the guy comes up, and he's a student in the class. Hi, I'm Robert Nicole. I'm the director of sequencing operations at the Broad Institute. And then I check and I find out that he personally was responsible for the kink in the curve that he was talking about. Okay? But the point here is that these changes in foundational technologies, tools, if you will, are part of what are enabling the advancement of the field that Mike's going to be talking about. And you have effects operating in a couple ways. One way is that if you look at the information being generated and archived, again, always nice when you have exponentials and you have vertical lines on an exponential graph. The amount of information being generated by sequencing right now is off the charts astonishing. And that information is providing a foundation for the development of modern synthetic biology. On the other hand, you also have changes in synthesis. So it's not just being able to do it more cheaply, it's also a question of being able to do more. And this gives you a sense of the length of the longest organisms or longest strands of DNA that are being published. And again, it's fast, it's happening very rapidly. So Mike's going to turn, because we've decided to switch it around, he's going to talk about political economy and the effects on global production and then into the definition.
Yeah, so this is a map. It's actually a little bit old. It's almost 10 years old now, just showing a survey of where these DNA synthesis facilities are. And you can see there's a lot of them in the US, but they're also all over the world, including in some places where we might think that some of these methods might be used to do harm. And so this, and again, this is very old, and I'm sure that if we looked at a newer map now, it would be a much, much greener map. And so the technology really is becoming widespread, and as Ken showed you in those previous graphs, it's really dirt cheap, and so it's easy to do. So the question is, where does the rest of synthetic biology come from? Well, it really has its birth in recombinant DNA technology, and I thought that just to follow up on the little panel discussion here where we were talking about unanticipated consequences. So recombinant DNA technology came out of some experiments where people were trying to figure out why is it that certain bacteria are resistant to infection by certain bacterial viruses. And that's what led to the discovery of restriction enzymes and our ability to take two pieces of DNA and basically splice them together to make a single molecule. And so synthetic biology, as it's currently defined, is really taking more of an engineering approach towards this particular technique. Okay, and the idea here is to have standardized parts that would then have predictable behavior, so that if I took part A and part D and part Q and put them together, I ought to be able to predict when those DNA molecules are introduced into a cell, and this could be a bacterial cell, it could be a yeast cell, it could even be a mammalian cell, can I predict how that behavior is going to occur? And so there's standardized parts, standardized technology, standardized nomenclature, and the BioBricks Foundation was built around this in terms of being a repository for these parts where people can get their hands on them and use them interchangeably, and presumably they will behave exactly the way that they're supposed to. So what are some of the non-military applications of synthetic biology? And I'm going to take you through a few examples so we could use this to look at disease mechanisms and see how we could change biological systems to make their behavior represent that of diseases. In terms of drug production, chemical production, one of the first applications of synthetic biology was the production of, or a new way of producing the anti-malarial drug that's commonly used. Biosensing, so can we use organisms to monitor hazardous conditions in the environment and such. And so here are some of those examples. So this is one where this group engineered E. coli, so basic bacterium, to produce Taxol, which as you all know is an anti-cancer chemotherapeutic drug, which is very expensive to produce right now, but if we could get a bug to make it, it'd be much, much cheaper. Another example here is this one of those biosensor types of things. So these guys engineered these bacteria to be sensitive to light. And then they also rigged the system so that the bacteria that were sensing light knew that they were next to bacteria that were not sensing light, and that they could then produce different edges based on how they equip those bacteria to engineer these different pathways. And this is the idea that I was talking about, where you're trying to come up with a standardized engineering type approach towards how these bugs are being manipulated and used, in this case, to make these nice photographs here. Now, one of the big advances really was this paper here, where this group at the Venter Institute showed that they could chemically synthesize a mouse mitochondrial genome. So here, they're basically using the technology that Ken talked about, just making a series of DNA molecules and assembling them together to make a large genome. So here we're talking about 16.3 kilobases, which is, at that time, a pretty big thing to make, okay, out of a series of 60 base pair long oligonucleotides, right? And this technology is now referred to as Gibson assembly. And in fact, most people now are using Gibson assembly to do their everyday cloning rather than using restriction enzymes, as was the original method for recombinant DNA technology. And so these guys took it one step further and actually synthesized a complete bacterial genome. They used the word creation, probably not the best choice of terms to use here. So basically what they did was to synthesize this entire bacterial genome, pull the genome out of a bacterial cell and replace it with this synthetic genome. And that genome allowed those bacteria to basically behave the same as the naturally occurring bacterium. And that's just shown here in a couple of photographs between the sort of artificial bacterium and the original, looking at it two different ways. And you can tell that at least morphologically, they're indistinguishable. And also then in terms of their metabolism, they were indistinguishable. And we don't need to go through the details of this map except to show that it's a very, very large genome. But again, they started with these very small molecules, assembled them all together to get 1 million base pairs of this bacterial genome. So the technology is there now where you can basically build a bacterium, at least at this sort of very simple level. So this is a very simple bacterium that they were using here. But you know, you could think about this is sort of the way that the field is evolving. And so there are some questions, I think, that this raises. And now maybe we could start moving a little bit more into sort of the ethical discussions. So here we go. It's a synthetic bacterial genome. It was transplanted into a pre-existing cell, as I said. And this was done using mycoplasma. So these are bacteria that don't have a cell wall, so they're specialized. They're a little bit easier to manipulate. And it's going to be a little bit more difficult to do this maybe with your run-of-the-mill bacteria, but that technology is probably not that far off. And so the question is, was this an incremental or predicted advance? Is this something that we should have been able to know would occur back when they first made that small mitochondrial genome, which was much, much smaller? So it's just a matter of time based on the improvements in the technology before this would occur. And then the question is, what is the applicability of this? So these guys, I think, did this as a proof of concept, but now what they would like to be able to do is, in effect, use a bacterium such as this as, if you will, a chassis onto which they can now add different components to make it do the sorts of things that they might want it to do. So sort of force this bacterium now to behave, again, in a predictable manner so it can be used for whatever applications there might be. And so I want to take you through a case study of how we think about some of the implications of these sorts of technologies, except that I'm going to do it with a non-synthetic biology example, and mainly because, number one, I was involved in this early on, and number two, because we thought that really this brings out all the different points, and it'll touch, I think, on a lot of the themes that we've been hearing about earlier today. So this deals with influenza virus H5N1, and again, it's not synthetic biology, but the question is, do we need to worry about experiments in which individuals are trying to benefit mankind, but now this might be misused? And then a corollary to this that we're going to come back to is, are these so-called gain-of-function experiments ethical? So is it ethical to take a pathogen and make it more virulent, right, is the question that we're asking here. But again, this can apply to other things besides just pathogens. So just a little bit of background. So first of all, the term dual use here in the life sciences is being used in a different way as has sort of classically been used. So for many, many years, we thought of dual use as technologies that have both a military use and a civilian use. And it was actually the Fink committee that Elizabeth sat on that came up with this new sort of definition for dual use, which is any technology that can be used for good or evil. And you can see that this vastly broadens what might fall under dual use. And I'm a member of a National Science Advisory Board on biosecurity that's been advising the government on the dual use issue and trying to come up with sort of a threshold, if you will, of when are we really going to worry about dual use. And that's how this whole involvement in the H5N1 situation came up. So H5N1 influenza is a highly pathogenic avian bug. So influenza viruses' natural host are birds. They're not humans. And so in birds, this virus is very severe, causes enormous economic losses to the poultry industry. And what was shown was that H5N1 was starting to infect humans in Asia, and people who got infected with this virus were not doing very well. So the case fatality rate was somewhere around 60%. So that means if you got infected, there's a 60% chance that you were going to die from this infection. And public health authorities were really worried, could this then become the cause of the next influenza pandemic? And just to put this in perspective, the worst influenza pandemic that we think about was the 1918 pandemic, and there, the case fatality rate was an order of magnitude less. Okay, so this would really, really be bad news if it could start to spread from human to human. And there was a big debate in the influenza community as to whether that might be the case. So some experts argued, no, it's not going to spread from human to human. If it could, it would have done that already. And then others argued, well, no, nature hasn't had the chance yet to try all the different mutational combinations that might allow for spread. And so a couple of laboratories decided to test this experimentally. Right? And so they basically did a simple experiment. They took the main molecule on the surface of the virus that allows for the virus to interact with the cell, called hemagglutinin, and they engineered it to be able to bind to receptors on human cells. So that's one of the big things that restricts avian viruses from infecting us, is that birds have different receptors on their cells for influenza virus than we do. And then they passaged it through ferrets. And ferrets are the accepted animal model for human infection with influenza virus. And what they got out were viruses that could be transmitted through the air from ferret to ferret, and then presumably from human to human. And so the question became, should these results be published? So these labs submitted the papers for publication. The US government found out about it and asked our advisory board, should these be published? And so the risk-benefit analysis looks something like this. So the benefits are, first of all, this says, yes, these viruses can become transmissible from human to human. Presumably, I mean, it is an animal model, but we wouldn't be using this animal model if we didn't think that it was really reflecting what was going on in humans. It gave us some basic knowledge on what are the determinants of transmission. So what sorts of mutations in what viral proteins need to occur for transmission to occur. And it's also important for surveillance efforts. So public health authorities can start looking for these particular mutations in birds and then know, hey, wait a minute, we really need to do something, for example, cull that flock so that the chances for that virus making the jump into humans might be lower. Now, the risks are, number one, someone can reconstruct this very deadly virus because they're going to publish the sequence of the exact mutations that allowed it to be transmissible. And number two, that type of work might proliferate, raising biosafety risks. And even though this work was done under very high-level containment labs, we know historically that there have been laboratory accidents quite frequently, more than we'd like to occur in these types of laboratories. And so, at the end of the day, what happened was that these papers were published, and the argument that sort of, I think, convinced most people that these results did need to be published was which was that these results needed to be out there for the public health authorities to be able to basically be on the lookout for these types of mutations. And the original recommendation that was made was to not publish the detailed information and just selectively share it with those who had a need to know. And the people at the NIH said, no, there's no way of doing that. And so basically then it became a binary decision. Either you let all the information out or you don't let any of the information out. And the decision was made to let it all out. So are there similar scenarios that we could think of with respect to synthetic biology that might be relevant to the military? And this is, just me sort of, using my imagination a little bit, although I, probably was informed by being on this committee and some of the things that we heard here. So, okay. So here's one idea. So I have a colleague at the University of Michigan who's in the neurology department, and he treats a lot of patients with chronic pain, and these tend to be cancer patients, right? And their cancer has metastasized, and it's really ugly. And so what he did was he engineered herpes virus to express this protein called enkephalin, okay, which blocks pain transmission. And the idea is to inject the patients with these herpes viruses. Herpes viruses naturally infect neurons, and now these neurons would be making this protein that would prevent them from sensing pain. Okay, so really, really cool technology, could help a lot of people. What if we turned that around? What if instead of producing enkephalon, we started producing neurotransmitters that cause pain? Okay, could this be used for torture? Is this legal under the Biological Weapons Convention? Right, so the same technology, dual use. Okay, what about this here? So here's another sort of imaginary example. So one state decides it's going to engineer bacteria to produce fuel for military vehicles, right? So that's one of the big pushes right now, is using synthetic biology to come up with alternative fuels. All right, but state Y might say, hey, wait a minute, we're going to engineer now a bacteriophage, a bacterial virus, that can specifically kill these bugs, right? So now, you know, our enemy's not going to be able to cheaply produce fuel for their vehicles and their planes. And now the first state comes back and says, okay, we're going to make our bacteria resistant to those bacteriophages. And so you can imagine this going back and forth and back and forth. And this reminds me of this book that I actually just read. I don't know if you guys, anybody's read this, but Margaret Atwood's a great author, and she's written this trilogy about a dystopian future. And this is the first book. And the premise is that it's this sort of biological engineering warfare among companies, not among states, that's gone awry. And I really recommend this book. It's really a very interesting take if you're interested in this whole idea of synthetic biology. Anyway, a couple of more examples. So what about trying to target individuals based on their genetic background? Okay, so there have been ideas to develop therapies that are targeted at specific genetic polymorphisms. So there might be individuals in certain groups who are more apt to come down with a disease, and you might want to come up with some sort of therapy that specifically targets those individuals. Well, you can imagine that instead of using a therapeutic gene, you could substitute a toxic gene, and now you can use this for malevolent purposes. Okay, so again, dual use. And then another example here would be vaccines. So one way to make vaccines is to attenuate a bacterium so that it's less pathogenic but still induces an immune response. But another way would be to try to perhaps enhance the bacteria. And this is another example of where you might get, say, an unintended consequence. So there was a lab in Australia that was trying to come up with a way to basically eliminate the rodent population, or to reduce the rodent population. And the idea they had was to take this virus called mousepox, which is a natural pathogen of mice, and it's in the same family of viruses as smallpox, and they wanted it to express this gene, which is an immune modulator. And they thought that this was actually going to allow for this virus to be better at sort of eliminating these mice. And in fact, it did, but what they also found then was that mice who were immune against mousepox, if you infected them with this virus, they would still become infected and die. So in other words, it made the virus hyper-virulent such that animals that were vaccinated or immunized were no longer protected against that virus. And so you can imagine, again, using this sort of thing in a human situation. And then the final example I want to give here is that you could think about things with respect to environmental cleanup, right? So what about bugs that eat pollutants? That would be great, right? So you could use those to sort of clean up the environment if you have an oil spill, something else like that. But similarly, you could engineer bugs that will eat nutrients, right? And then use that for agricultural types of warfare. And you can imagine the economic outcomes of that. So again, there are all these different applications or potential applications of synthetic biology that are all really, really good, but it wouldn't take much to turn them on their head and really use them for to do harm. And so finally, there's this issue of creating life that I want to come back to. And so can we really build designer bacteria to perform a designated function that we want? So build it from scratch the way that the lab, the group that was at the Venter Institute did this, right? And the question is, how predictable is synthetic biology really going to be? Because again, we're taking engineering principles, and remember, with engineering principles, you know, if you have a screw, that's a certain type of screw, it's going to fit into a certain type of bolt, and that's always going to be the case. If that screw is built to those specifications, and that bolt is, that nut is built to the specifications, those two are going to intermesh. How predictable is a living system going to be when you've got to take into account mutation, and then there's going to be some sort of selective pressures that might be put on there. And so, and so the system may evolve in a way that's unpredictable. And so that's something that we need to keep in mind as we're approaching the synthetic biology. And then I'm going to turn it over to Ken on this final note, which is engineering a pathogen to be more virulent ethical? Right? So is it legal under the Biological Weapons Convention to make something that's more pathogenic? I don't know. I think, it could probably be argued either way. And then should the military be performing gain-of-function experiments? In particular, if that does somehow get us close to that boundary of violating international treaties, right? So are certain types of human enhancements being done by using biological systems or synthetic biology? Are those going to be sort of creating new weapons in effect, okay? And so I think that's something that we need to think about. We know, for example, that the human microbiome, which are the normal bacteria that inhabit our bodies all the time, can affect our behavior, can affect our health and wellness. What if we said we could take a warfighter and change their microbiome such that maybe they're going to be stronger, or they're going to be more alert? Some of the things that Bob was alluding to earlier this morning. Are those types of things ethical to do? And then again, is any kind of thing where we're enhancing the function of even a bacterium, that's a gain of function. And at what point do we say gain of function is going too far because we really don't know what the behavior of that organism is going to be down the line? And so now I'm going to give it back to Ken to investigate these ethical questions.
Okay, so Mike laid out a series of cases that are hair-raising. In fact, even my hair is starting to curl after listening to Michael go through those examples. And before we can talk about ethical and legal issues associated with those examples and others, let's take 5 minutes on ethics and law for dummies. I look around the room with great trepidation, noting that we're sitting in a moot courtroom and discussion of law by social scientists should probably never take place in law schools. But the basic point that I want to make is that as we were deliberating as a committee, we were often dealing with tensions or conflicts across the frameworks that we were using for evaluation. And we'll hear words like law or ethics tossed around, but oftentimes it sure isn't clear what we mean by it. And everyone, of course, believes that everything they do is sitting in the sweet spot in the middle of these Venn diagrams, but it isn't so. There are powerful psychological reasons for wanting to believe that everything that you do is ethical from a deontological and a categorical perspective and legal, and also serving the national interest narrowly defined while also benefiting all of humanity. And our capacity for reshaping our conceptions of ethics and law to make that so is probably the only force more powerful than Mike's conception of synthetic biology. So let's go through very quickly into some of the sources of conflict or ambiguity associated with each, a little bit on applications to synthetic biology, and then we'll turn to Ruth's favorite approach, which is stories. And we're going to go through some stories of good and bad examples of engagement with ethical and legal issues in synthetic biology, sometimes with headlines leading the way, and in other cases, maybe in advance of disaster. So beginning with law, simple point, very, very straightforward. If you're really turning to law, to statutes or international conventions, to customary international law, to case law, the basic problem that we have is that oftentimes there are conflicts of laws, points of tension across these sources of legal norms. And those tensions, those controversies often mean that in areas of emerging technologies in particular, I would argue, we're often left with weak guidance from existing bodies of law for emerging technologies like synthetic biology. So a couple quick examples. If you take existing law here, turning to international legal conventions, you have the Biological Weapons Convention, and then to implement the Biological Weapons Convention, you have this very nice agreement cutting across lots and lots of countries, not including China and others, called the Australia Group and their guidelines. So this is where life is supposed to be good. Countries get together, they decide what they're going to worry about. And the guidelines and US policy, the select agent list, consist of a list of organisms to worry about. Now the problem is that synthetic biology, as Mike pointed out, is doing all kinds of weird stuff that doesn't necessarily fit the list, some of which could be dangerous, but it just doesn't happen to be listed. And indeed, the whole approach to defining biosecurity by listing known pathogens of concern is probably not sufficient to some of the cases that we're going to be dealing with in a couple moments. It's a problem. Or you take US policy on dual-use research of concern. And that policy, which we'll go into in 2 minutes, really also consists of a list of experiments and a list of organisms of concern. Frankly, my friends around Cambridge have come up with applications of synthetic biology that are very dirty, dual-use-y, with serious security implications that unequivocally do not fit under the guidelines. And we've been doing a little experiment by actually tweaking and poking and prodding the government agencies that are responsible for regulating in this area. And the response we're getting is, yeah, that's something to worry about, but it's not covered. And we'll talk about those examples in a second. The fit between existing law and emerging technologies is often highly irregular, and we'll go through a little bit more on this. Deontology. Now, who am I to talk about deontology sitting at Notre Dame University? All right? But we could be using sources that range from religion to natural law and then applying them to try to assess the morality of specific actions in terms of generalized rules. Controversies? Lots of them. Even within the Catholic Church, there are on occasion some controversies over ethics and law. Certainly when you go across religions, there are. I mentioned that at the drone panel, Father Hare was talking about Catholic just war theory as his basis for interpretation. Others would be arguing in terms of prohibitions on killing that would be even broader. Still others were saying, I don't like this just war theory. And each was actually trying to draw on sources of ethics and law from different religious strains or secular sources. And they didn't agree. And of course, that's not surprising. But there's also conflict. If you accept a principle, do not kill innocent civilians, then we have this little issue of how do you define civilian and innocent? Or do not torture. We all agree as Americans, do not torture. Is waterboarding torture or specific other categories of action torture? And a deontological approach is probably a lot easier and clearer in terms of its application than what we're about to talk about, the consequentialist. But even here, there's ambiguity. In synthetic biology, if you're talking about modifying or creating artificial life forms, what do you mean by artificial life and creation? Again, J. Craig Venner and his guys regretted the use of the word creation in that article, except for Venner himself, who liked the idea of getting the publicity. Engaging in discriminant targeting of racial or ethnic subgroups. Again, there are deontological issues that are raised by that. You could argue that coming up with means of targeting or doing harm to genetically defined, racially specific subgroups is something that may be just plain wrong. If we're talking consequentialist approaches, life gets even more complicated. We're talking here about an evaluation of actions on the basis of observed outcomes, looking back, or expected outcomes. Given the scope and mandate of the committee, we're talking here a little more about expected outcomes. As you're sitting there doing your research, what is likely to follow from the research, and what culpability or responsibility do you have? So what do people fight over? Well, if you're looking at the effects of something looking back, the credibility of the observations on effects is often called into question. Do you believe what is being reported? But if you're talking about the future, expected effects, life gets even worse. You have models, implicit or explicit, that you're using to generate outcomes or to attribute causality. And we're talking about models of expected effects in a world where significant uncertainty and controversy and complexity is associated, particularly with fields like synthetic biology. And as Ruth pointed out in her remarks, when you're talking not so much about just the evolution of the basic technology, but applications, human uses, it gets extremely complicated. And it's not because of bad modeling. Okay? I want to take exception a little bit to the assumption that Dan Lindley made when he was remarking, saying, we can just do a better job forecasting. This is not a question of having slightly better methods. There are fundamental epistemological problems in terms of being able to generate high-quality models on the uses or applications of technologies. At the end of the day, you don't know. And then there are obvious problems. Valuation of observed and expected outcomes across countries. Are you doing it cosmopolitan or are you doing it national? So in synthetic biology, all of these factors, uncertainty and complexity, trade-offs across what you value, and then the valuation of national versus international effects are really significant issues. In fact, with emerging technologies, the consequentialist approaches become both really important because you want to be able to tell what effect they're going to have, but to be honest, you really have to be, again, coming up with approaches to evaluation that are a little bit more humble and with heavy emphasis on gathering the information to improve the models in an atmosphere where people are not necessarily going to believe what's being presented, and the issue of credibility is something that we're going to come back to in our examples. So if we turn from that to obligations, now the first point that I want to turn to here is to put Mike on the spot. I'm a social scientist. I don't develop bugs. Mike does. So Mike, if you could talk about your duties and responsibilities.
Yeah, so this is a great question because this is one of the things that even in the sort of 10 years that dual use has been discussed in the life sciences has become pretty evident to me, which is that many individuals who are researchers in the life sciences don't necessarily, in my view, understand their duty in terms of thinking about the possible consequences of their actions. And so, for example, one of the arguments that came up again and again during this whole influenza controversy was that, look, I'm trying to do these experiments to help mankind. I want to be able to help the public health authorities. I want to be able to, figure out, what different changes to the virus are going to affect its transmissibility and such. It's not my problem to worry about whether someone's going to try to use this to do harm. And I think that that's really a false way of thinking about things, because I think we do have to consider the potential implications of our actions. And I think that especially in the scientific community, that we need to realize that we're doing this research for the public. And if we can't on the one hand say we're here to help the public, and on the other hand say I don't need to consider the risks to the public of doing that. And so I think the scientific community really has to really embrace its duty more. Other scientists would just say, this is not my problem, I don't even want to have to think about it. Now, I have to tell you, to be honest, my impression is that the good news here is that at least in the synthetic biology community, those people have been very, very attentive to this issue of the potential for risk. And I think it's because they understand a lot of the things that Ken and I have been talking about with respect to what the unknowns might be, and that this is a new area. We don't really understand how things are going to behave, and so we are going to pay more attention to this. But I have to tell you that scientists, life scientists in general, are really not attuned to these sorts of ethical issues. So just one final kind of anecdote, which is that I'm involved in our research responsibility and ethics course for our graduate students. So all of our graduate students have to take the course. And at the beginning of each session, I ask the students, how many of you have heard about the Biological Weapons Convention? And I can tell you that I've been doing this course now for, I want to say 7 or 8 years. I usually do 4 or 5 sessions a year, 20 students each, so you could do the math. In all those years, I've had 2 students raise their hand and say that they've heard of the BWC.
Where were they from, Mike?
Yeah, so one of them was not a U.S. citizen, actually. Yeah. So this is the thing that we're faced with is we have a whole generation of biologists who don't know the environment in which they're working.
And so we've been actually trying to educate biologists. One case that I'm not going to go into here, but just 2 sentences. There's something called the International Genetically Engineered Machine Competition, and it has 3,000 students, 200 plus teams from all over the world. And in addition to teaching the technology, we've been making a systematic effort to train people on their obligations and duties to be evaluating and managing risk. But this is with a strange group of partners: the UN Biological Weapons Convention Implementation Support Unit, the Federal Bureau of Investigation, and others. We've also been trying to assume a little bit of responsibility for potential danger by screening every project and by screening all parts in the registry. SGI DNA volunteered its services with Archetype, a pretty good tool, to be screening every biological part in the registry for function and for organism of origin. And we're trying to do that in part as a model of reasonably good practice. Why? Because you have a whole bunch of kids running around that don't necessarily know about what's expected. And we don't want people showing up in Mike's class at the advanced graduate level not knowing what their obligations are, not knowing how to define infectivity and pathogenicity, not knowing what's legal or illegal, not knowing what their duties and obligations are. Now, let's take what Mike and I have put forward as a general obligation or duty to take account of these concerns and considerations and translate them into particulars. So, in concrete terms, we're suggesting that there are 4 things that people ought to be thinking about. And we're ordering it in terms of elementary to advanced, from introductory to graduate level, 101, 201, 301, and 401. So at a very basic level, of course, you want to be evaluating the fit between the experiments and research that you're doing or funding and existing law, norms, from both perspectives, but all this based on existing knowledge. So you take the best available information, you give it your best shot. At a minimum, that's what you ought to be able to do. But oftentimes, as noted earlier, existing information will not be sufficient. So we're suggesting that you have a second obligation, which is to identify potentially critical gaps in knowledge and do your best to fill them. Now, if you're an individual researcher, there may be limits on what you're able to do. If you're a research funder, we are suggesting that research funders who are putting in support for H5N1 research, NIH, Tony Fauci for particular, would also have an obligation to be funding research on the potential implications. And we'll go through some examples of research funders doing exactly that. You also have a bit of an obligation, and this will be a little more controversial. After all, you're just a scientist. Why should you have to do this? But you have a bit of an obligation to try to identify potential gaps or limitations of existing policy and law, and to spur debate on those gaps as you're moving into an area. Not all researchers will accept that as a duty. George Church of Harvard has, and so have others. And finally, humility again. Recognize that after you do your best on 101, 201, and 301, you're still going to screw it up. So the issue of planned adaptation and learning and self-correction and openness to the new information is something that we believe in as critically significant given the failures of forecasting. So following Ruth's admonition, too much of this poetry, it's time for stories. Let's go over 3 cases, 3 stories that can serve as illustrations. So the first case, case 1, is actually giving the denouement of the H5N1 story that Mike started earlier. And if we turn to this story, it's actually kind of fascinating. Okay, I just love that too, the sneezing ferret and the sneezing human. Okay, so these 2 articles came out. Mike was actually on the committee that was trying to decide what the heck to do with these articles. And as you can see up there, there were a whole series of repercussions that followed from the fight over is it ethical, is it right for this kind of research to be done. Now, a couple little observations here. There was a process for handling the problem, for evaluating the risks and the ethics. Mike was part of that process. And at the end of the day, NSAB accepted publication with a split vote on one and unanimous vote on the other. So just a little note before we jump into more complicated aspects of this case. Why was there a split vote on one and unanimous support for the other? And because Mike, I don't think, is supposed to comment on the precise allocation or details of voting, what I've heard, not from Mike, but from other members, is that the Kawaoka version was working with a weakened version of H5N1, and the methods being used to do the modification were just a little bit more responsible. That Fouchier, otherwise known as the Belgian cowboy in biological engineering circles, was working with a full-strength strain using methods that almost anyone could use to produce the result. And the concern among members of the committee, and the reason why there was a split, was that people really felt that the danger associated with what he was doing was great, and the attention to methods for mitigating and managing risk was too small. So that's all particular to H5N1. Now let's move to the larger implications in cases 2013 and 14. So in 2013, the World Health Organization got involved. Well, they were involved earlier, but there was this massive international informal consultation. For those of you who've done diplomacy, informal consultations mean that people can actually talk. Informal consultations mean that it's controlled by protocol only, with official statements being read. This was a fascinating meeting with intense divisions between Americans and Europeans over how to interpret what is a matter of security concern. In fact, I walked away deeply depressed, as Europeans were viewing American concerns over security as manifestations of American paranoia, and Americans were viewing the Europeans as excessively and insanely risk tolerant. Now, this is a reversal of the usual story on risk tolerance. But there were other aspects of this that I found deeply disturbing, and I want to highlight them. One was that, well, in fact, with Herb, we were sitting at a conference on dual-use research at a university which will remain anonymous. And the director of a national laboratory, who will remain anonymous, said, all of this can be handled very easily. And you know what his suggestion was? Take H5N1 and other gain-of-function research, take vaccines development associated with this kind of research, and just give it to us. And we'll do it. And we'll do it properly, behind closed doors, in classified domains. And he said, we can do it because we have reliable personnel, we have controls on the distribution of information, we understand what the laws are, we'll take care of the problem. Now, I actually couldn't stand it, and I will probably never get a job at that particular laboratory. But the issues are several, right? First, there's a little issue of personnel reliability within laboratories if you go back to the anthrax case. But lay that aside. The real issue here is that if you take that experimentation and conduct it in a national laboratory, you could argue easily that it's a violation of international law under the Bioweapons Convention. The development of gain-of-function, more virulent and transmissible H5N1, together with a vaccine, pushes the limits of the convention. And then if you do that in classified settings, how do you reassure others that our intentions are pure and that we're not pushing the edges? It becomes very difficult. I don't know if you agree with that or not. The other issue, though, is more general. And it comes back to a theme that we've discussed earlier. To what extent is it possible to have good evaluations of information on ethics and consequences in classified settings? Again, this is not just a rap on the military. Because it's also true when you talk about pharmaceutical companies operating behind walls of proprietary information, or a hospital worried about malpractice and hence bottling up information. In general, how do you have honest discussion under conditions when information is sealed, when participants are limited? And I would suggest that the worst, or one of the bad ways of doing or handling the DIRC issues would be to move it behind classified settings and do dangerous stuff. Okay, other notes. Oh yes, the OSTP policy on oversight of DIRC, 2014, released this week. And it lists dual-use research of concern, types of experiments, and pathogens. And NIH isn't supposed to fund this stuff, and everyone's supposed to be really careful, and no one's supposed to publish, and you're supposed to monitor and report. I just want to note that at least one really interesting, and I mean interesting, experiment. A category of applications with potentially serious security implications really doesn't fit. And it was an interesting experiment because we have a manuscript under review, 2 manuscripts under review at a journal, one on the policy effects, one on the technology. And the journal did what they were supposed to do. They shipped it off to, not NSAB, but to the OSTP. And, you know, the response came back, yeah, there are security implications. This is not covered by the scope of current DIRC policy. The reason why the scientists in question were submitting the piece for review early, and this was actually before all the work was done, was because they hoped to spur debate on the implications of the work that they were proposing in advance of doing it. They felt it was their ethical and moral obligation to get the information out to spur debate before the applications were done. And they wanted the policy piece to go with it, and I'll discuss it further once it's published, but not before, we can't. I also want to note that even in this H5N1 area, there's still a lot of controversy over what the benefits were, and part of this, again, is a question of who's allowed to participate in debates. A gentleman at the Pasteur Institute, whom you know, is still hot and bothered, claiming that the benefits associated with the research were grossly exaggerated, and he might be right. Okay, let's move on to a second case. Security risks of DNA synthesis of potentially dangerous organisms. Now, this is one where the prompts, well, okay, the prompts are headlines, okay? I mean, this is not the kind of headline that you want if you're running an agency. You don't want a headline on anthrax, certainly that's Fort Detrick's problem. You also don't want the headlines on 1918 Reconstruction being dangerous, or this little one at the bottom. How many of you are regular readers of The Guardian? No. Oh, you are, okay. So here you have a British publication that is far more responsible than the worst, but they also have a real flair for investigative reporting. So they got a team of reporters to go around and order fragments of smallpox from DNA synthesis houses. It was not the whole smallpox genome, it was just little bits. And in fact, the task of reassembling those bits into smallpox would be quite formidable. But it was a little disturbing because guess what happened? They placed the orders for delivery to private houses, and the orders were filled. Hence the article, Lacks Laws, Assembly of Deadly Virus. So they're exaggerating a little bit, but here's the good news. What happened as a response to that combination of anthrax and the Spanish flu recreation and the Guardian article were that you had a series of studies. Now, we've all seen studies in government. Ruth has probably read 190 studies that went nowhere in the last year alone. What's interesting about this case is that the recommendations in the studies actually led to something. Okay, they're saying there's danger here because of synthesis. What do we do about it? And what happened was an interesting arrangement where HHS came up with guidelines or recommendations on what to worry about, and then 2 private transnational screening consortia formed, and they're screening all of the orders that go to the companies that are members of the consortia. For the customer, are they weird and dangerous? And for the sequences that are being synthesized. And it's a system that's working reasonably well. And there are adaptive elements to it as well, in the sense that they're systematically trying to evaluate threats to the regime, possible changes in industrial structure, new technologies, and to evaluate the adequacy of what they're doing to manage the risk. In my view, this is self-interested conduct because they don't want to be dinged, and it's also ethical conduct. They're making serious effort to engage with risk in a manner that's actually working reasonably well so far. Final example. The final example here deals with a non-headline case. It's a lot easier to get action after people have been burned. In this case, what I find remarkable is that a funding agency is doing something good before getting kicked in the teeth with newspaper headlines. And that agency is actually the National Science Foundation. So NSF, Cellular and Molecular Biology, Environmental and Engineering Directorate, plus SINBRG, And by the way, that's a miracle anytime that you get more than one piece of any organization working together. Together with EPA and Sloan, have been putting resources in to engaging with environmental implications of synthetic biology before they get nailed, before the headlines come in. And so we've been working with them on this, all right? And this is a case where the Wilson Center in Washington and our little group at MIT have been putting on a series of workshops, taking examples of synthetic biology that are either near application, nearly done, occasionally stretching a little bit, and then working through and evaluating the risks. What do we know about the environmental implications and effects, and in some cases, security implications? And what we did was we started working through all of those, starting with a sensor, then moving on to E. coli, a chassis, then moving on to cyanobacteria modified to produce sugars, and then finally to a whole bunch of stuff in the last year. In fact, 2 months ago. So, for example, on biofuels, we had biofuels firms coming in and presenting on their approaches to manufacturing biofuels and where they expected to be in a couple years. You don't do it just on what they're doing right now. And they were careful not to reveal too much proprietary information, but they were pretty straightforward. The reason why they were straightforward and wanted to do it was we also had the gentleman sitting there from EPA. He's the guy that writes the licenses. And they were all anxious to be sitting there with a guy who would ultimately be deciding on what would be allowed out. But to have a pre-regulatory discussion on the risks, they had an interest in being nice. But what was also amazing, if you look over there at that slide with all the people, some look interested and some look bored. Okay, the idea of having a diverse group together, scientists and engineers to be sure, but others, I think they're called stakeholders. So you see Sarah Carter, Jimmy's granddaughter, but also JCVI, J. Craig Venner Institute, sitting next to Eric, the leader of the campaign against synthetic biology, Friends of the Earth, next to JD, the Technology Center's leader of their campaign, next to Steve Evans, Dow AgroSciences, and next to him, Gwen McClung from EPA. Now, I mention this because you might assume that it would be impossible to have a civil discussion of risk under the conditions of, you want to talk about slightly diverse backgrounds. What happened was a miracle. They disagreed over some things, to be sure. The NGOs, of course, wanted precaution, and the firms, of course, wanted, don't bother us, we want to just do it. But they agreed on what the risks kind of were, and more significantly, they agreed on areas of uncertainty. And they all favored, unanimously, all favored government funding, public funding, not private sector funding, for a research program to address and identify potential environmental consequences, to identify or flag concrete areas of uncertainty and fill the gaps. So then the second miracle that happens is that NSF funded exactly what you might hope for, which would be an effort to define research agendas after flagging areas of uncertainty. And so they put in a couple $100,000. We would gladly accept larger contributions to do another set of workshops that used different applications, but the focus was on generating RFPs, helping shape their funding priorities. And the fact that the funders accepted a duty or responsibility, and right to protect, nah, a right to know, a right to investigate, a right to understand. And that duty was, we used several funny examples, okay, a nitrogen fixating non-legume, a gene drive for editing DNA in wild populations. That funny little plant that you see down there is actually out there right now. Kickstarter, you've heard of Kickstarter, the novel fundraising approach. They were giving away seeds for luminescent plants for investors in the plant. And then a biomining application that's really quite exciting. So the applications were in and of themselves fascinating, but the good part was that it used or generated a research agenda, and there was buy-in on doing the research. And the Europeans are tracking along the same ways right now. So what we have is, again, a surprising amount of interest in the synthetic biology community, not everywhere might, but in synthetic biology, in identifying gaps and filling them. So finally, unresolved issues for discussion, and I'm going to hand the microphone to you for this part. If we turn and look, at the end of the day, who's really obligated to act on these ethical and legal concerns? How early or late, and how? And the report speaks to these issues, but I think it's useful to be reconsidering in light of some specific examples. What institutions have a real interest in taking these interests seriously? Plenty of folks are willing to play lip service. Who's going to put their money and skin into the game in terms of actually funding research and seeing that it's carried out? Or are you going to sit back and say, we're going to just do it on the basis of existing knowledge, we'll give it an honest shot? And then finally, the issue of classification in particular and secrecy more broadly, what effect does it have on ethical deliberation? You know, we're making an effort to engage with these issues in the report, in our work in synthetic biology, but these are hard questions, not easy ones. And as Herb indicated in his opening remarks, it's the start of a dialogue.
I think you should keep that because I suspect we'll have questions and you'll probably have to answer them. Gentlemen, if you would both. Please, the floor is open. Oh, yes, and thank you. I got so wrapped up. Thank you both. It was excellent. I like the presentation style. It was dynamic, wasn't it?
Okay, comments, questions?
We have 10 minutes,
10
I'm
minutes,
told,
okay.
by our fearless leader.
Again, we can also do the Oprah, and I can kind of hand the microphone to people as well. Comments or questions? Okay.
That was a great talk. I especially liked the concrete obligations part because I think many times the technologists are the ones who, I mean, they're the best, in the best position to identify the gaps that we have actually. And I think taking that on as a technologist is an important role, and they're the ones that are hitting this stuff first and can help define the uncertainty areas, I think you called them as well. So I completely support that.
So this is a great story that the scientists who are involved in synthetic biology actually figured out they needed to take it upon themselves to worry about this stuff.
Not all of them.
Not all of them. I guess it's a two-part question. What, if anything, other than just recognition of the fact that it was dangerous work, was there anything else? Was there anything that gelled them to be doing that? And I guess, second is, how do we translate this to other areas of research? Are there ways in which we can sort of catalyze that kind of behavior among other research groups?
So let me try to tackle the second question first. My impression has been that the younger generation of scientists is much more open to thinking about these things and becoming aware about these things than my generation is. And so right now, that's my best hope, is that the people who are growing up hearing about these things more and being asked to think about them are going to think about them, whereas people who are set in their ways are really not going to change. With respect to whether there are other things that, I guess, what do you think, Ken? The whole DIY thing, right, probably spurred that. So the do-it-yourself movement, where people were trying to basically do biology, in their garages or in these, incubator types of things and stuff like that. I think the people in more academic labs were worried that this may taint, the whole situation. So I think they felt like, it's our obligation to make sure that we're setting a good example, and then potentially those individuals will also follow along, and to a large degree they have been. And Ken mentioned the FBI has been doing a lot of outreach to these groups, and it's really been well received, and so I think that's really worked out well.
I mean, just one other note. I think people in this field looked at the example of GMOs in Europe and said, if we mess this up, what we're doing could get shut down. Genetically modified organisms and their regulation in Europe. The other thing was that you had NGOs that were campaigning to basically ban commercialization of synthetic biology, some of whom were again at our meetings. And the fact that they were actually campaigning, not frankly really expecting to secure a moratorium, but the fact that they were campaigning, I think, actually whetted the appetite or interest on the part of the companies and the academic researchers in engaging with the risk. But part of it was also, I think, just a culture of responsibility among leaders in the field. So if you have the fifth-rate clown running around saying we have to be attentive to ethics, it's not going to work well, but when George Church is sitting there making the appeal for engaging directly, and he's putting his work on the line behind it, I think that that also helped as well. But there are plenty of things that are going on, but it's an interesting story for sure.
Yeah, so, during the whole H5N1 controversy, there were never any leaders in the scientific field coming out and saying, we really need to stop and think about this stuff. Everybody was just dead silent, except for the people directly involved. And so I think that really, tarnishes the discussion.
Well, there's a reason.
There's been, actually, Elizabeth had her hand up, Herb has hand up, and then Ruth. Elizabeth, do you still want to be on the floor? She gave away your microphone.
Yeah, there's a reason, by the way, why those scholars were not willing to talk very much critically about H5N1 gain of function. Who funded that research? NIH. Who personally sat behind it? Fauci. Would you really want to get the principal source of funding in your research area mad at you by mouthing off? And that was one reason why people were not talking, Mike, and people have told me that after voicing concerns over what was going on.
Well, I have a great deal to say, but I know others do too. But let me start by saying that serving on the Fink Commission was really very interesting, as I recall. That was about an 18-month exercise. And we started out with, I thought, really quite a bit of hostility on the part of the biological research community, and they shifted over that time. It was fairly impressive to see. This story, however, is very heartening. That said, I wonder, in a minute we're going to talk about cybersecurity. There seems to be quite a different perspective in that community, and one wonders why and what's the difference. And then finally, I have two other comments. I found myself thinking about medieval times and how science gets so far out ahead of the popular culture that this disconnect then creates part of the problem. And I think that may have been the GMO situation in Europe, and it probably goes on here in different ways, as you know. But now here's more of a substantive question. I'm on the board of the Public Interest Declassification Board, and we have been just struggling mightily, coming out with reports which no one reads, unlike this report, saying, here's some things that should be changed and so on. Can you give some thought to this? I think we just have a classification problem that is huge. We're generating information faster than we can, even think about it, and we've got just years of backup, and we've got a system that is kind of, what shall we say, first in, first out, that has nothing to do with priority or significance, and really is I think, disadvantaging us in today's world, where since the end of the Cold War, we've had just a seismic shift in what's important. Well, we're busy declassifying things in the Reagan era, or if that, it's just so dysfunctional. So what can you do there?
Let me take Herb's question as well, because he's been patiently waiting, and you can speak to both of them. Is that all right?
My comment is on a completely different topic than Elizabeth. So why don't you?
Well, we actually only have 3 minutes left because.
I'll ask that question offline then. That's fine.
See, he's a science guy, so I'm going to put him on the spot. Go ahead.
So, I think the question of classification, I think the real issue. I think that the issue, to some degree, comes back to the one that Ken raised about this list-based system of kind of what's bad and what's not. And I think that's where we kind of fall into traps of what information should not be out there and what information is good to be out there. And then I think layered on top of that here, of course, right, is people have made arguments about, First Amendment rights to publish their results, academic freedom, all these sorts of things that I think really sort of, are distractions from the real question, which is, what is the ethical thing to do? What's the right thing to do without getting bogged down in these, sort of more legal things or list-based things and really coming up with a more normative approach is what I think is going to be the most useful thing.
So earlier in the panel before this one, you were talking about how classification often proceeds as you go from general to specific, from basic to applied, and particularly to really applied. And the same could be true here in the sense that if you take the application of a general principle, debate over that should take place in the public domain for sure. Okay? So you take gain-of-function research, and there should have been an open debate over gain-of-function research, not restricted to NSAP, frankly, but much broader. Again, acknowledging or pointing to the fact that the breadth of that debate would be essential because so many actors were fearful of making statements that could reduce their funding. Okay. Now, here's the trick, okay? So no one's going to be arguing necessarily about development of a weapon with a specific design as something that perhaps should be restricted. But it's in between where life gets difficult, and there aren't going to be any bright lines. In general, I would err on the side of not classifying. And part of this is just based on seeing too many examples of how secrecy has often really led to bad evaluations of technology and effects. But there's no magic bullet. Herb's suggestion in the report that we get around this problem by trying to create communities of people who have clearances, who kind of get together and is diverse enough, all well and good, but someone has to actually really take seriously the problem and structure a process where you have diverse points of view reflected. And in fact, the temptation to stack the deck in terms of the evaluation so that it comes out the way that you want is something that I think would probably be present.
Do you want to give time for one more question?
Actually, it's just,
Would you like this?
Yeah.
very quick comment, as much as a question. It sounds like in this field, a lot of things were done well. Could this be used as bouncing the framework against what has been done, looking at whether all the necessary stakeholders were engaged? What can we learn from this as it would advance the cause of the framework?
So it's not perfect by a long shot, but I actually have been shocked at how well it's been working. I mean, I never thought that you get those guys in the room and they'd agree on getting the funding and then NSF would give them money, okay? And it goes a little beyond that. Proposal in right now for a cooperative threat reduction grant. To do what? Engagement and education in regions of interest, out of IGEM, the educational activity. Does it travel is really your question. And I'm not sure. If I look at cyber, okay, the characteristics of the field, it's very different. The fact that people are actually doing the bad stuff already, the fact that you're already relying, well, there are analogies that are similar in terms of transnational networks doing much of the management of threat, internet exchange operators versus the synthesizers. But cyber is so huge with so many actors that it's almost hard to get a handle on. And synthetic biology is not tiny, but we could actually identify 25 people that would be the acknowledged leaders in that field everywhere in the world without much difficulty. And they would still have influence through their laboratories on setting values and ethics as well as methods. So I'm not sure. I'd love to see some of this stuff.
Be a test bed
Yeah.
for the framework to see what scales and what does not, what translates and what does not.
No, I agree. Mike?
The only thing I would add is that, synthetic biology sort of grew up with this as part of its culture, right? Whereas cyber, again, I think the technology outpaced.
But synthetic biology is probably not the last domain to grow up.
It's not. It's not. And it could be a model. It could be a model. Until until something bad happens, then it becomes the negative model.
Actually, at this, I think we have to end now, but I would like to draw on something you've been talking about to point out something I think is a wisdom I'm not certain you all completely appreciate that you've already put in action. By linking a successful case within questions of larger cyber emerging weapons, you force people, as you pointed out, Elizabeth, to look at why certain success stories exist in ways that can be modeled in areas that haven't yet figured that out. And I just want to compliment the wisdom here in your whole panel for putting these and interlinking these, as you pointed out this morning. I think that's a step towards the future that you're looking for. What are our applications? So let's give everybody here a hand. And thank you, Dan Rozek.